Chapter 20 is about genetic variation and evolution. And so genetic variation is really cool. We all have genetic variation in this class. Unless you have an identical twin, everybody in this classroom is genetically different from each other. And that's true for other organisms as well. This is the multicolored Asian ladybird beetle. They're not called ladybugs because they're actually beetles, not bugs. And this is just one species, and they have a ton of color variation on their wings. And they have over 200 different color patterns that they can display depending on what their genes are like. And it's regulated by one gene. This difference in color, 200 different color variations, is regulated by a single gene in this organism. So what causes genetic variation? Mutations. Mutations cause genetic variation. And there's two different types of mutations we can have in our body. We can have somatic mutations, which are in our body cells, so like your skin cells, or your heart cells, or your liver cell, or your red blood cells. Those are your somatic cells. If you have a mutation in your somatic cell, it's just gonna stay on your body. So it's just gonna affect you. If you have a mutation in your germline, which are your sexual cells, so those are the ones that you use for reproduction, so for mammals, our sperm and our eggs. If you have a mutation in your germline, you're gonna pass that mutation on to your offspring. We have a lot of mutations in our body. So if anybody gets cancer, it's because you had a mutation happen in your cells. You're not going to pass that cancer on to your child if it's in one of your somatic cells. Because it's just in you, you're not passing those cells, that DNA, on to your offspring. If you have a mutation in your germline cells, that's when you transmit it to your offspring. So for our ladybugs, those 200 different color variations, that occurred because there was a mutation in that gene for color in a parent's either sperm or egg. That's where that mutation occurred. There's different types of mutations. You can have a harmful mutation. So that's something that actually hurts the individual. So for us, an example of a harmful mutation in our somatic cells is mutations that cause cancer. There's neutral mutations, and we have a ton of these in our body. They're not good or bad. They just happen, and it doesn't really affect anything. And then you can have advantageous mutations, and those are ones that improve the carrier's chance of survival. So it helps you do better in the environment that you're living in. How do mutations occur? Think back to 150.
Like overdivision of a cell?
So you could have overdivision of a cell. Yeah, so there's lots of different ways mutations occur in our DNA. They're random. That's the most important thing. Mutations are random. If you are a squirrel and you're living outside and all of a sudden it becomes really snowy all the time and it's way colder and you have brown fur, are you going to develop a mutation for your offspring to have white fur so they're less likely to get eaten by a hawk? No. The mutation is just going to occur randomly. You cannot force a mutation to help you be better adapted for your environment. The second thing that helps with genetic variation after mutations happen is recombination. So that's when you're shuffling mutations around during mitosis or meiosis, or during meiosis, and new combinations of these mutations occur. So two things cause genetic variation: mutations, recombinations. Mutations are random, they happen by chance. So when we're thinking about genetic variation, we need to think about different groups of organisms. So a species is a group of individuals that can exchange genetic material through interbreeding, aka through reproduction. But not all organisms we're going to talk about this semester do reproduction in the same way, so we're going to call it interbreeding. So humans are a species. Mosquitoes, certain mosquitoes are a species. Those ladybird beetles are a species. Goldfish are a species. If they can share their DNA through reproduction in some way, they're a species. A gene pool consists of all the alleles present in all individuals in a species. So this map is showing different groups of humans, and each color represents a distinct allele or set of alleles in these populations of humans. And this is actually looking at alleles on Y chromosomes of males. And the different colors, you can see that these groups over here are more likely to have the allele that they colored in red, where people over here in Asia are more likely to have alleles that code for blue color. So we can see that even though we know these are all humans, we can all interbreed with each other and make offspring, the genes that we have are different across regions where we live. Because people living in Europe are more likely to mate with each other. People living over here in Asia are more likely to mate with each other. But you can have somebody from Europe come over here and mate with people in here, and then you can have mixing of different alleles. Because we're all the same species, we can all mate with each other. A population is within a species. So that is an interbreeding group of organisms of the same species living in the same geographic area. So if you're on the island of Great Britain, that's a population because you're all together on that one island. If you're on the island of Japan, that's a population because you're all on that one island breeding together. So different things keep populations in check, and we'll actually talk about that next week, what makes a population a population. So we can measure this genetic variation using math. Yay, math's so fun. So we can look at the allele frequency, which is the number of a particular allele that is present within a certain population, divided by the total number of alleles. So mathematically, we could say allele frequency is equal to the number of the copies of the allele we're interested in, divided by the total number of alleles in a population. So we're going to look at these flowers for peas, because we spent a lot of time last semester talking about Mendel's peas. Let's talk about the flowers associated with these peas. So we have two phenotypes. We have purple flowers and we have white flowers. The purple allele is dominant to the white allele. So we're looking at these three flowers. And I want to know, what are the frequencies? What is the allele frequency for the big W, the dominant allele, and the little w, the recessive allele, if every flower in our population is white? So if we only had white flowers in our population, what is the allele frequency for big W, and what is the allele frequency for the little w? Let you think about that. You could talk to your neighbors. It's not a trick question. I know some of you are like, wait, what? Oh, we're so quiet. Okay. So if we only have white flowers, what is the allele? What alleles do you have to have for white flowers? Little w, little w. So if we only have white flowers, what's the only allele we can have in our population? Little w. So what is our allele frequency for little w's? 100%, because that's all there is. So if we look at our copies of little w's, if we only have little w's, it's 100%. What's the frequency for big W? 0, because it doesn't exist in this population. So that's why we want to look at allele frequency. So we could say there's actually no genetic variation in this population of white flowers. We're never going to get a purple flower unless a mutation occurs. So now let's look at one that's actually a little more challenging. So this is when we actually have a population. Here's our population of pea flowers. And we have 1, 2, 3, 4, 5, 6. We have 6 purple flowers that are homozygous dominant. We have 1 heterozygous purple flower, and we have 2 homozygous recessive white flowers. So you need to figure out what the allele frequencies are for W and W. I'm going to let you think about that for 15 seconds. See if you could figure out the answer before we go through it together. All right. So if we want to start with the big W, we want to use this equation. The big W frequency is equal to the number of copies of big W divided by the total number of alleles in the population. So the first thing we need to figure out is how many alleles are there in the population? How many total alleles? So how many individuals do we have in this population? 9. How many alleles does each individual have? 2. So how many total alleles do we have in the population? 18. You're halfway there to the answer. Yay. So we have 18 alleles in our population total. Now we need to figure out how many big W alleles we have. So how many big W alleles do we have? We have 13. So you can count 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13. So we have 13 alleles. You just divide those two numbers. You get 0.72. This is a good time to tell you need to have some way to calculate numbers for class on Thursday. So like your cell phone calculator is fine. We're not doing anything harder than, I think the hardest thing is a square root. So our allele frequency for purple flowers is 0.72. Now what is our allele frequency for little w? 5 out of 18, because there's 1, 2, 3, 4, 5 little w's, and there's the same number of alleles because it's the same population. 0.28 is our frequency for little w. Something really important when you're calculating this is that your allele frequencies should always add up to 1, because if we're thinking about frequencies, we want 100% of our population, so 1. So a great way to check your math on these problems is 0.72 plus 0.28 equal to 1? Yes. So at least you did your math right. Hopefully. So we know that 100% of our population is now represented in our allele frequencies. All right. Another example problem. This time you're given the genotypic frequencies. You're not given the population size. So in this example, 0.36 of our population has homozygous dominant alleles. 0.48 of our population has the heterozygous genotype, and 0.16 has the homozygous recessive genotype. What are the frequencies? Ooh, this one's harder, I know. And just fair warning, there's going to be a sapling question on this. So you should really pay attention to this. So if we want to find our frequency for big W, which is our number, we'll take this equation, the number of our homozygotes plus half the number of our heterozygotes. Because we know that our homozygotes only have that dominant allele, and our heterozygotes have half, right? They only have 1, not 2 of that dominant allele. So if we're looking at our big W, we have our 0.36 plus half of 0.48, because only half of that frequency for heterozygotes is due to that dominant allele. And then the same for little w. And remember, your allele frequencies should always add up to 1. Okay, so we use half here for our heterozygotes because they only have 1 of the copies of the allele we're interested in. They don't have 2, so we need to take only half of their percentage, or their frequency. Any questions on how we measure allele frequencies? If you have a question and you're too shy to ask it, write it down right now so you can email it to me. Okay? All right, so we're going to now switch. We're still talking about genetic variation, But now we're going to talk about the Hardy-Weinberg equation. If you've heard of this before in other biology classes, you've probably heard Hardy-Weinberg equilibrium. We're not talking about that yet. We're just going to work on using the equation right now. The Hardy-Weinberg equation predicts genotype frequencies from allele frequencies. You actually can use this to go the other way as well. In the Hardy-Weinberg equation, the dominant allele we use the letter P. For the recessive allele, we use the letter Q. I don't know why, that's just what somebody decided a long time ago, and that's what we use. So if we're thinking about how we calculated our allele frequencies last time, on the other slides, I said allele frequencies always have to equal 1, because we're talking about 100% of our population. So thinking about that, our dominant allele plus our recessive allele frequencies always have to equal 1. So we could say P plus Q equals 1. And if we want to think about, I switched from flowers to bunnies. If we want to think about our bunnies, very similar to our pea flowers, but we have brown bunnies and white bunnies. Brown is our dominant allele, and then little b is our recessive allele for white. If we're thinking about a population of bunnies, there's 3 potential genotypes that the bunnies in our population can have. So if we're thinking on a population scale, we want to think about all the eggs and all the sperm that can be given by all the individuals in that population. So if we think back to last semester and look at our Punnett squares, there's 2 alleles that are possible that a bunny can potentially give. A big B, that dominant allele, or a little b, the recessive allele. So if we're thinking about all of our female rabbits, they could give the big B, which is the same as saying P, or a little b, which is the same as saying Q. The same is true for all the male bunnies. In their sperm, they either give a big B or a little b. Or, if we're thinking about Hardy-Weinberg, they can give a P and a Q. So what's going to be the genotype of this particular offspring? Big B, so they're homozygous dominant. Or if we're thinking about Hardy-Weinberg, what are we going to say? PP. And what does PP mean mathematically? P squared. So if you are an individual that has the dominant, homozygous dominant genotype, in Hardy-Weinberg, we say you have P squared. What's P squared? I'm going to tell you in a little bit. But it just means you have 2 dominant alleles. Now, if we're heterozygous, so we could be this offspring or this offspring, what is our genotype going to be? If you're heterozygous, what's your genotype? Big B, little b. So if you're a big B, little b in Hardy-Weinberg, we say that you're PQ because you have 1 dominant allele and 1 recessive allele. And then what is our Hardy-Weinberg number going to be for our homozygous recessive individuals? Q squared, because we have QQ or Q squared. So if we're thinking about our genotypes, we have our homozygous dominant genotype is big B, or P squared. Our heterozygous genotypes are big B, or PQ. And then our homozygous recessive individuals are little b, or Q squared. So our Hardy-Weinberg equation then is all of the different genotypes you can have put into one equation. So we have P squared, so that's our homozygous dominant individuals. 2PQ. We have 2 PQs because there's 2 opportunities for us to have a heterozygous individual. So PQ plus PQ is equal to 2PQ. And then we have our homozygous recessive individuals, our Q squared. Because we're talking about our entire population, this is equal to 1, because we're talking about all the genotypic frequencies, every individual in our population. For those of you in the back, can you see this down here? Yes, no? Okay, I'll try to move stuff up a little bit and not put it so low in the future. Okay, so this p squared plus 2pq plus q squared equals 1 is your Hardy-Weinberg equation. This is probably one of the few things you will remember from this class because it's this weird equation that just sticks in your head for the rest of your life. You'll be like, Dr. Parker taught me that. So that's our Hardy-Weinberg equation. So we can use this to figure out our genotypic frequencies, and we can use p plus q to figure out our allele frequencies. So those are the two equations that we want to be using when we're doing Hardy-Weinberg problems. So we're going to do a practice problem. Yay! Still with our bunnies. And just a reminder, this is just from the last slide. So just what these different things mean, what P and Q mean, what our equations are. So I'm giving you an allele frequency. So we know in our population of rabbits, this is our fur color. And we know from doing testing that the allele frequency for the dominant allele, big B, is 0.82. The question that you're being asked is, what is the genotypic frequencies for these rabbits and what is their phenotypic frequencies? So the first thing you need to do is figure out what the allele frequency is for P. So you have P here, 0.82. Yay, that was so hard, right there. So what is our allele frequency going to be for B? How do we figure that out? Yeah. Perfect. They need to equal 1. P plus Q needs to equal 1. We know that this is P, so 1 minus P, or 1 minus 0.82, is 0.18. So our allele frequency for our little b, or for Q, is 0.18, or 18% of our population have the little b allele. So next we need to figure out what our genotypic frequencies are. So here we want to figure out for a homozygous dominant. So how are we going to figure out our homozygous dominant genotype frequency? Somebody other than Savannah. Yeah. Almost. So we want to use, we know P. So if we have big B, all we need to do is square P. Bless you. So big B is equal to P squared. So we want to square our allelic frequency. So 0.82 squared is equal to 0.67. So 67% of the individuals in our population are homozygous dominant for brown fur color. How are we going to figure out our heterozygotes? 2PQ. So 2PQ is how we figure out heterozygotes. So 2 times our P, which is 0.82, times Q, which is 0.18, is equal to 0.3. So 30% of our individuals are heterozygotes in our population. How are we going to figure out homozygous recessive? Q squared, 0.3. Is there another way you could have figured out our homozygous recessive genotype other than doing Q squared? Yeah. Perfect. So if we know what our P squared and 2PQ is, we can figure out Q squared by setting it equal to 1 and solving for Q squared. Because we're talking about our entire population, we want it to be equal to 100% of the individuals. So if you did 1 minus 0.67 minus 0.3, you would get 0.03, or 3%. Now, how are we going to figure out our phenotypic frequencies for brown fur color? Any ideas?
Can't you just like add like the
Yeah, perfect. So we would just add all the genotypes that get us brown fur color, which is our homozygous dominant and our heterozygotes, or p squared plus 2pq, which is equal to 0.97. I didn't put the math on there because it went off of the slide. So I hope you can use the numbers here. And then how do we figure out white? It's just little b because there's only one phenotypic frequency that can be for white, so it's just q squared. Any questions on how to use Hardy-Weinberg to measure genetic variation in our population? No, okay. And I know I just went through the math, but I'm going to explain mostly on Thursday why we use this, but a little bit today. All right, so evolution. So it's great that we know we have genetic variation, but why does genetic variation matter when we're thinking about evolution? Because all that evolution is a change in allele frequency in a population over time. That's all evolution is. So if we have these really cool beetles, in our first generation, here's our allele frequency. We have 97% have this big A allele. We'll say it codes for black coloration in our cool beetles. And then over many generations, now we have our 10th generation. We now only have 0.6% of our population with the big A allele and 0.4% of our population with the little A allele. Evolution has happened in our beetle population. That is what evolution is. It's a change in allele frequencies in a population over time. It doesn't mean that these beetles now have unicorn horns and tails. It just means that their allele frequency has changed. So now we have more individuals that have that little A allele. Any questions? So that's why we use Hardy-Weinberg, because of Hardy-Weinberg equation helps us see if there is change in our allele frequencies, because we can measure that using numbers. And that's how we determine if evolution is happening in our populations. Evolution is simply a change in allele frequency in a population over time. There are agents of evolutionary change, and we will talk about them on Thursday. This includes genetic drift, non-random mating, mutations, gene flow, and natural selection. So any of these things can be acting on a population, and it could be changing their allele frequency. The Hardy-Weinberg equilibrium, which is used in the Hardy-Weinberg equation, describes a population that is not evolving. So there's no change in allele frequencies. So if you took population in the first generation and a population in the 100th generation, you use that Hardy-Weinberg equation, there would be no difference in your allele frequencies. That means evolution is not happening. Things that help a population not evolve: large population sizes, random mating, so you just mate with whoever, it doesn't matter what their phenotype or genotype is. There's no mutations in the population. There's no migration, so you're not having new organisms coming in. And there's no natural selection. This rarely happens. If you're in Hardy-Weinberg equilibrium, there is no evolution, no change in your allele frequency. Everything is going to stay the same in your population. Your allele frequencies will stay the same. So in this example with our green beetles, if you haven't figured out, I really like insects, we're going to keep having green beetles. No evolution. Our allele frequency for the green color is the same over generations. we're going to skip that question. All right, so we're going to look at if our population is evolving. So we're back to our brown and white bunnies. We want to know if our population is in Hardy-Weinberg equilibrium. Here are your genotypic frequencies here. So our homozygous dominant is 0.45, our heterozygotes are 0.25, and our homozygous recessive are 0.3. Okay, so these are our genotypes in our generation, and we want to know if our population is in Hardy-Weinberg equilibrium when it comes to fur color in rabbits. So the first thing that you're going to do, oh, there's also a sapling question on this, so you should probably definitely pay attention. First, what we need to do is we need to figure out what our allele frequencies are, okay? So we know our genotypic frequencies, but we need to figure out our allele frequencies. So remember, sorry if you can't see this in the back, our allele frequencies, when we know our genotypic frequencies, is equal to the number of heterozygotes plus 1/2 of our, oops, did I say that backwards? Let me say that again. Our allele frequency is equal to our number of homozygotes plus 1/2 of our heterozygotes. So if we're looking at our dominant B allele, it's going to be 0.45 plus 1/2 of 0.25. So our allele frequency for B is 0.575. We've already done a practice problem like this, but now we're putting it into the context of Hardy-Weinberg. So then you're going to do the same thing for our recessive allele, little b. 0.3 plus 1/2 of 0.25 is equal to 0.425. How do we check to make sure our math is right? It needs to add up to 1. Do we add up to 1? Yes. I did my math right. Now, what we need to do next is knowing our allele frequencies. If these are our allele frequencies, what is our genotypic frequency supposed to be if we are in Hardy-Weinberg equilibrium? This is when we want to use our Hardy-Weinberg equation, because this equation, when it equals to 1, tells us that we are not evolving. So we've already done this. To find our genotypic frequency for big B is the same as p squared, so 0.33. So if you take 0.575, do p squared, you get 0.33. And when we're doing math, if I ever have the wrong number on here, please tell me so I can fix it. I'm human and I make mistakes. For heterozygotes, we need 2p squared, so 2 times 0.575 times 0.425 is equal to 0.489. And for our homozygous recessive individuals, we use q squared, so 0.425 squared is equal to 0.18. Now, looking at these genotypic frequencies and these genotypic frequencies, is our population in Hardy-Weinberg equilibrium? No. For our population to be in Hardy-Weinberg equilibrium, we need to have these genotypic frequencies match these genotypic frequencies. They need to be the same. So that's one way to test to see if our population is in Hardy-Weinberg equilibrium. When you say no, that means there is something acting on the population that is causing evolution to happen. We have a change in allele frequencies. If we were in Hardy-Weinberg, we would have these frequencies represented here, but they're different. Any questions? No. Okay. We're going to skip that one. Yeah. All right. So one of the things that can affect evolution or causes evolution to happen is natural selection. Natural selection is one of my favorite things to talk about when we think about biology. So the conditions required for natural selection is you need to have variation among individuals in your population. And we're going to talk about natural selection in lab this week as well. Some of your variation must be inherited, so you need to be able to pass that variation on to your offspring. Survival and reproductive success, so how many offspring you have, vary among the individuals based on that variation. And survival and reproductive success is tied to those traits that you get from your parents. So if we think about an example with butterflies, because, bugs are cool. Here we start with our pink butterfly. We have a mutation that creates variation. So now we have 3 colors. We have blue, pink, and red. Say these butterflies like to land on red flowers. If you're blue, you're going to stick out like a sore thumb. That's an unfavorable environment for a blue butterfly. It will be eaten by a bird and die. It will not have any offspring. It is removed from our population. This is natural selection happening. We have heritable variation. Survival and reproductive success is different among our individuals. This one died because it wasn't fit for its environment. Reproduction and mutations continue to occur. So now we have our pink butterfly, and now it's a different pink, and we have different shades of red. Favorable mutations are more likely to survive over time, so we're on our red flowers. There's no more blue, so the pink butterflies stick out way more than the red butterflies on the red flower. So only the red butterflies survive and reproduce, and over time, we now went from having pink butterflies to red butterflies because there's unfavorable conditions in the environment that allow some organisms to survive better than others. We're going to talk about how this works a little bit more on Thursday. But that is how natural selection works. It makes sense. This is where survival of the fittest comes from. If you are fit, you do well in your environment, you survive, you reproduce. Your offspring have those traits that help you survive in the environment. They're more likely to survive and reproduce, and so on and so forth. So natural selection is the same as saying survival of the fittest. Another example where we see natural selection play out that actually affects us in a big way is evolution of antibiotic resistance in pathogens. You take an antibiotic when you have a bacterial infection. That antibiotic kills most of the bacteria. The one that are able to survive that antibiotic reproduce in your body, and they survive. They gave that resistance gene to their offspring. That resistant bacteria is able to be passed on to other individuals, and then you have things that are resistant to our medicines, like antibiotic-resistant tuberculosis. That's an example of natural selection happening in our own bodies. That's why if you're sick and you take antibiotics, you should take the entire prescription so you could kill as many of those organisms as possible.